You're listening to Idea Collider, a show that explores the world of asymmetric learning. On this show, I will sit down with pharmaceutical experts and business leaders to discuss how to embrace uncertainty and the different learning style that follows. I'm your host, Mike Rea. Let's get into the show. Welcome to another podcast. Delighted to be interviewing Sekar Kathiresan from what was Verve and now Lilly. As you'll see when we get into the conversation, there's so much great experience to share from Sek, building, starting biotech, what it looks like when you move that biotech into a large organization. And one of my favorite subjects clearly is how you commercialize something that the world hasn't seen before and what that looks like and the kind of opportunities that come from thinking differently about that whole journey. I know you'll enjoy this. Sek is one of the gentler and more thoughtful folks that I've had on here, and real pleasure to talk to. Okay, Sek, fantastic to have you on Idea Collider. I know we've tried for a while to make this happen, and to have this post, what you guys have done with Lilly is fantastic. So welcome to the podcast. I'd like to start with just getting to know a bit more about you, Sek, because really you've had your early life journey, from India through to really being a cardiovascular disease pioneer is pretty compelling. Do you want to tell us more about your origins and story?
Yeah. So I was born in India, in southern India, Tamil Nadu, and I came to this country in 1980.
Mhm.
My father had come to do his PhD in mechanical engineering at the University of Pittsburgh in 1975. And then he brought my brother and I kind of 5 years later to the US after he had finished his PhD. And so I grew up in Pittsburgh, elementary school there, high school, and then college at University of Pennsylvania, where I majored in history, and then went to Harvard Medical School in 1992, and then did almost all my clinical training in Boston, first training, and then ultimately was on staff at Mass General Hospital as a cardiologist as well as a human genetics researcher. And then we'll get into a bit more of that story, but about 10, 15 years into that journey as an academic physician, a physician scientist, I founded Verve in 2018 and then have led it since. So that's been kind of my journey to the United States and then researching cardiovascular disease.
A history to medicine, is that a kind of normal thing for people to bridge?
Well, my favorite subject in high school was American history. I had a wonderful teacher that really got me excited about that topic. And so I kind of pursued it in college, actually always knowing that I would do medicine and that I would have the ample time and opportunity to really go deep into medicine during medical school and beyond. So I wanted to use the college years to kind of broaden out a little bit, and that's kind of how I landed on history.
Yeah. And then to start Verve Therapeutics, I mean, that's an interesting, step from medicine into founding a company. What was what was your goal at that point?
Yeah, so, my background, I kind of think about my career in maybe like 3 chapters,
Mm,
so the first chapter is really learning to be a doctor and caring for patients.
mm,
So I did that, for about 10, 12 years where, did clinical training and then cardiology fellowship. I was a chief resident at Mass General Hospital. And really at the end of all that, I was preventive cardiology and I led that group within Mass General Hospital in Boston. So that's kind of the first chapter. The second chapter is really learning to be a scientist.
sure.
And so after I finished my clinical training, Mike, I spent about 5 years as a postdoctoral research fellow in human genetics at the Broad Institute and Mass General Hospital and focused on the genetics of heart attack risk as well as resistance to heart disease. And so that's really the second chapter. And the learnings from that second chapter really led to what's now the third chapter, which is to develop a medicine, to treat cardiovascular disease, leveraging the learnings from that research time. And that all started in 2018. And so that's kind of the chapter we're in now. And really the proximate event that led to that shift from researching to kind of entrepreneurship was really a grant competition from the American Heart Association.
Okay.
And so, I started my research lab in 2008 and done a fair amount of work on the genes that might confer, as I said, risk or resistant to heart attack.
Yeah.
And in 2016, the American Heart Association said, "Hey, we want the best ideas to cure heart attack, cure coronary heart disease."
Mhm.
And this is the problem I've been working on my whole career. And so I said, "Oh, I think we have some ideas around this." And so we put in an application. So this was going to be a $75 million grant
Wow.
for one person,
Okay.
really attractive for
Yeah.
any
Absolutely.
academic to apply to. So the idea that we put in, Mike, is actually what eventually became Verve. It's
Yeah.
the idea of a one-time therapy, lifelong cholesterol lowering, and we can get into kind of how we arrived at this as a potential solution to heart attack maybe in a minute. But we put this application in with this idea and we didn't win. And somebody else got it. And then, we're quite disappointed. But we got a couple of people excited about it. One was Google Ventures. And so I had a couple of colleagues who had a role there. They thought, oh, this is a big idea and we could get behind this. So this is again 2016. So
Mhm.
we spent 2 years, Mike, meeting weekly, incubating the company, developing, the initial people, the resources, and also IP necessary. And then in 2018, launched with a Series A. And shortly thereafter, like in 2019, early 2019, I decided to leave academia altogether and come and lead the company full time.
Mhm.
And that's really my transition from research to entrepreneurship.
And your leap into leadership, I mean, that's a big, bold move in itself. I mean, did you take your medical training to another level then? Or, what was your belief in yourself that you could do that job?
When we started in 2016, the company was just, again, pure science, right? The idea, at least, meaning that, we spent, as I said, about 10 years researching the genes that might increase risk or confer protection. And the fundamental insight that came from all that research work was that if your blood cholesterol was really low lifelong,
Mhm.
it's actually very hard to get a heart attack.
Yeah.
Okay. And we learned that because we found a bunch of people who naturally had their cholesterol low lifelong.
Yeah.
Okay. And they had these gene mutations that would turn off a cholesterol raising gene in the liver. As a result of that, these people who have lifelong low cholesterol would be remarkably protected from heart attack. And so that's what gave rise to this idea that, hey, if we could develop a medicine that would mimic that natural situation, we might actually have a treatment, if not a cure, for heart disease.
Mhm.
Okay. So that's really the science that led to the company, the idea. As I said, we spent 2 years incubating. So in 2018, Mike, we're sitting around, we had a board. I was still, I had just actually been promoted to full professor at Harvard Medical School. I was like super excited about the research and what we had accomplished. But then we started this company and we closed the Series A. We had GV, we had F Prime, we had Bob Nelson from Arch,
Yeah.
Biomatics, a really stellar group of funders. And our first board meeting, and we're looking at each other, and people are like, "Who's going to lead this thing?" You know, and so I kind of was like, "Well, maybe maybe I should put my, the hat in the ring." And so I kind of tested the idea with a couple of people, including Krishna Yeshwant, who's the lead at GV. And Krishna, they were pretty excited about the possibility, even though I had no prior,
Yeah.
you know, business experience.
Yeah.
They were excited about, my ability to potentially lead this thing.
Mhm.
And so that gave me a lot of confidence. And so I decided to, as I said, jump all in and transition. And I remarkably in the first couple of years, Mike, found that the skills that I had learned in academia running a research lab
Yeah.
actually translated fairly well for the first couple of years of the company. Meaning that what we had to do in academia was, pick a problem, an important problem, develop a strategy to solve the problem, raise money, and get good people.
Mm.
Okay. Well, it turned out those were the four major things you had to do when, starting a science, foundational, biotech, right?
Yeah.
Preclinical. And so that's kind of the initial journey.
That's excellent. And I mean, but you were essentially looking for a moonshot, with this kind of gene editing technology because, before you, this kind of, statins, PCSK9s, everything was there. But your belief in this kind of gene editing approach was, I guess, from your scientific career into a business case that, well, let's explore the business case as well when we get to it. But, was Google fascinated by your moonshot, or was it a straight business answer for them?
No, absolutely by the moonshot. In fact, the AHA competition was called One Brave Idea.
Okay.
And that's kind of what they were looking for. And, that's what we turned into Verve. And I think what GV was attracted to was if this worked, this is the answer to heart attack,
Hmm.
would be the answer to heart attack. And I had deep conviction that this was the answer to heart attack based on the genetics. And maybe let's dive in a little bit into that science that kind of gave me that deep conviction. As you said, we have options right now to lower cholesterol. And if you think about heart disease, coronary heart disease, so this is the blockages in the heart arteries that lead to heart attack, the leading cause of death in the world, still,
Okay.
this is the problem of over time of cholesterol basically building up in the wall of the artery, leading to ultimate clogging of the artery. And if it clogs completely, then you end up having death of heart tissue, which is a heart attack.
Yeah.
Okay. Now, there's been about 100 years of research on this problem. And believe it or not, even before the human genetics, a lot of it pointed to cholesterol as being the key driver.
Yeah.
Now, for me, what nailed it as cholesterol being not only necessary, but sufficient, low cholesterol being sufficient to solve the problem
Hmm.
was this search of the genome that we did, unbiased search of all the genes in the genome. And we said, all right, what might protect people from this disease? And we found 8 things, Mike, 8 genes where there were mutations that protected people from heart attack.
Hmm.
And believe it or not, all 8 landed on cholesterol. They were all cholesterol genes. We went into this without any prior hypotheses, and all 8 genes were mutations that turned off a cholesterol raising gene in the liver and led to low levels of different cholesterol carrying particles in the bloodstream.
Hmm.
And so this is what gave me the conviction and the confidence that if we could develop a medicine that mimicked that natural situation, and the CRISPR technology that just had come on the scene in 2012 gave us that possibility of a one-time therapy, turning off a cholesterol raising gene in the liver with CRISPR-Cas9 editing, and then mimicking the natural situation. We were able to really marry these two, right?
Hmm.
That was a bold, brave idea.
Yeah.
And I think, again, GV is interested in this kind of thing because they want to change the world. And that's, I think, what led to them latching on and kind of supporting our initial concept, even though
Yeah.
most people thought it was crazy.
Yeah.
And even American Heart didn't believe in us, right? They didn't want to fund this thing.
Right. I should ask actually who won the competition in the first place.
Yeah, that's a story for another day.
Okay. I assume they're not part of the next big company out there. Okay. I mean the kind of conviction and the confidence that you had to pursue, you say CRISPR-Cas9, novel technology, a one-off shot at, essentially editing someone's gene for the rest of their lives. What kind of boldness and bravery does that take to kind of say, we're going to do this and it's going to be great?
All these things, as you know, kind of start with concept in a moment, right?
Mhm.
And then you're given some resources, and then it's up to you to show that, hey, this can be done.
Yeah.
Okay. And I think in 2016, when we initially proposed the idea, we're about 4 years into even this idea of CRISPR and
Yeah.
what it could do. Incredible amount of excitement. And then there were offshoots of CRISPR, and we actually ended up leveraging a technology called base editing, which is essentially an improvement on CRISPR, standard CRISPR, which just cuts.
Yeah.
Technology we're using doesn't cut DNA, but rather it's like a pencil and eraser and switches one letter, an A, for example, at a single spot to another letter like a G at that spot. And we leveraged this technology to make a change like that, a spelling change that would turn off a cholesterol raising gene. In our case, for the lead product is the gene PCSK9. Now, the base editing technology was invented by somebody named David Liu at the Broad Institute and had just been shown to work in cells and was starting to do some work in small animal models. And that's when we got access to the technology. And so we had a couple of different technologies to accomplish this kind of one-time therapy, lifelong cholesterol lowering, but none of it had been shown to work basically in mice or
Mm.
larger animal. Now that's kind of the idea. And then the moment was, I think, a moment of tremendous enthusiasm and for biotech and biology, right?
Mhm.
You were part of that and saw it. There was a tremendous run, roughly from 2010, 11, 12,
Yeah.
all the way to probably 21, where there's a tremendous amount of capital flowing to new ideas and largely fueled by the zero interest rate policy of the time. And, and we were able to ride that. We basically secured about 60 million in funding for Series A. And I think at that point, with all that capital flowing, platforms and new big ideas were actually what was in vogue. And that was us.
Yeah.
And so we were very grateful for that. But then I think what has allowed us to move further is our focus. We were very focused on a problem, a very specific problem. We knew what we wanted to accomplish. We were agnostic or, you know, to technology. We weren't wedded to any single technology.
Mhm.
So we said, all right, we want to turn off PCSK9 gene in the liver. We want to use some editing approach and we want to deliver safely
Mhm.
to the liver. And so we looked at a couple of different delivery technologies, and in 2016, 2017, already at that point said, "We're gonna do lipid nanoparticle delivery system," because and for a variety of reasons, but we didn't think that the other technology, virus,
Yeah.
would scale or be cost effective enough or maybe even safe enough to reach large indications. On the editing side, we were open to the standard CRISPR-Cas9, the cutting. We were also open to this new approach, base editing. We got access to both of them. And what we did in the first couple of years, Mike, of the company was evaluate in cells, mice, non-human primates, all these different possibilities.
Mm.
And then we were the first to get base editing to work in large animal models, particularly non-human primates, in 2020. That data allowed us to take the company public in 2021 and get the requisite capital to take the molecule, our first generation molecule, to patients in 2022. So if you think about it, that story of starting the company in 2018 with a brand new technology idea, and then in 4 years having getting first patient dosed
Mm.
was really that rapidity, that rapid pace was all due to the focus, I would say.
Yeah.
That was probably our secret sauce in the first few years.
Yeah, and it's kind of fascinating. I mean, you're describing this kind of classic Edison-like, pursuit of innovation, right? Which is solving problems, but towards a goal which is clearer than I think most pharma companies would have for, an early phase asset.
That's right. I really do think that our intense focus on these kind of 3 major carriers of cholesterol,
Mhm.
and we had a target for each that we had picked out from the human genetics. And the first, as I said, was PCSK9 for LDL. The second was ANGPTL3 gene, the second gene for triglyceride-rich lipoproteins, and the third was this gene called LPA for lipoprotein(a).
Mm.
So that was all from the beginning. And we said, all right, we're going to focus on lipid nanoparticle delivery. We don't know which lipid nanoparticle system to use.
Yeah.
We surveyed the world. We got access to a few, tested them. And then, as I said, on the editing side too, we were quite open. And, it's not to say that this was a straightforward kind of a linear, up and to the right story,
No.
and we can get into that. But there are lots of kind of ups and downs as well, even in the few years. But we were always anticipating what the risks could be
Yeah.
and had often had a contingency for the risk. And then a couple of times that contingency saved us.
Yeah. I mean, we can get into those ups and downs. I'd be interested also in your approach to the clinic, because not many people had done what you did. In terms of lifelong gene edits into phase one, phase two, phase three studies. So, how did you, as a team, begin to think about getting this towards approval?
Yeah, so we had very compelling preclinical data,
Hmm.
right? When we got to about 2020, 2021. And as I said, there was a lot of excitement about that data. We published it in the journal Nature in 2021. But it is a big step to go from that
Mm.
to a first human being dosed with this
Yep.
new modality. And there, there was a lot of work done in terms of regulators, right,
Yep.
around the world. And it was a shared learning experience, I would say. For example, with the FDA, the US FDA, I believe we may have been the first to go in front of them with an in vivo gene editing product for the
Yep.
liver.
Yeah.
And so we took our lumps as a result. I think people often use the phrase innovators tax.
Hmm.
I think we paid that, because they, were unfamiliar with this approach. They didn't know what they wanted in terms of even preclinical data,
Yeah.
risk or assess risk. And there was a fair amount of back and forth in terms of educating each other, And, as you may remember, or you may know, when we put in our initial application, we were put on hold. And, in terms of additional requirements that they wanted prior to us being able to dose patients in the US.
Of course. Yeah.
Meanwhile, we had taken a global strategy because we didn't know which regulator was going to ask for what.
Hmm.
So we'd gone to other countries as well, UK, New Zealand, and actually had gotten clearances in those other countries. So in fact, our first patient dosed was in New Zealand in 2022.
Okay.
But that moment of dosing that first patient with this new type of medicine was really, I think, an awesome moment, in terms of just thinking about we felt a tremendous amount of responsibility, but also a lot of gratitude that somebody is willing to volunteer to take this kind of new approach, with the potential benefit to them,
Yeah.
but most people were volunteering because of altruism,
Yes.
because they thought that their kids could benefit or their humanity at large could benefit. So that's what I remember from July 22 after getting the call from New Zealand saying that first patient had been dosed.
Yeah. Fantastic. And it's interesting because, by that point, are you able to change anything after that, or really you're set and, locked and loaded for what you're taking to the regulators?
Well, so that was our first molecule called
Mhm.
Verve 101, and we did test that in in about a dozen or so patients. But as you know, it worked. We had got human proof of concept that you could edit,
Mm.
turn off a cholesterol raising gene. We're the first to show that base editing can work in patients, and shared that data with the public in December 2023, about 2 years after year and a half after that first dosing. But we had some safety challenges with that initial acute safety with that initial lipid nanoparticle delivery system in terms of LFT changes and platelets. And so we had a backup molecule, a second molecule called Verve 102 that switched out the delivery system, kept the guide and the editor, the gene editing machinery as the same, but had a different delivery vehicle. And we had developed this almost in parallel because we knew that delivery was a risk, and so we wanted to have a contingency
Mm.
as, you know, as part of, just good drug development practice.
Yeah.
And it turned out that, ultimately we had to pause that first program in 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 2024, April 2024, and literally the next month we were able to transition to Verve 102, the next gen molecule with this delivery system, different delivery system that had a targeting ligand called GalNAc added to it. And that's actually the molecule that now is in the clinical development, and that's the molecule we generated data for and shared in phase 1 data in April of 25 this year, and ultimately led to the acquisition by Lilly.
Yeah, okay, and we'll get onto that kind of acquisition. And I'm interested because of where you started as, as a physician moving into and running a pharma company, where does the ethical sort of dimension of, what you've done, come in? 'Cause you are pioneering a lot of, ethical approaches for our industry, right?
Yeah. I mean, I think there are 2 aspects of it that I'd like to talk about. One is when people think about editing, there's editing your own body,
Mhm.
that you get consent for, and we can talk about that. And that's very much like other procedures that we give consent to
Yeah.
medicine, There are lots of procedures, right, particularly in cardiovascular medicine
Yeah.
that are one time, irreversible, so permanent, intended for lifelong benefit. The best example is like bypass surgery,
Mhm.
or a coronary stent procedure, or, ICD implantation, or AFib ablation. So there's a range of procedures that people give consent to. So there's that, and that's kind of what our approach is. It's basically a procedure. It's less a drug, more like a one-time molecular surgical procedure.
Yeah.
Okay. And so that's that. But then there's the other issue that people have, which is, might you pass on this edit to your children?
Yeah.
You know, and so editing the germline cells, either sperm cells or in men, or egg cells in females, and that kind of editing could end up, if those cells get edited, then you might give rise to kids that would carry forward that edit.
Mhm.
So that second issue is a big ethical no-no. There's a consensus in the field, and we've looked very closely to make sure that we are not editing egg cells or sperm cells. And when we infuse our drug into the bloodstream, which is how it's delivered, a one-time intravenous infusion over about 3 to 4 hours, we've looked extensively, and we do not edit egg cells or sperm cells. So we can put that issue aside.
Yeah.
Coming back to what it really is, it's like any other, as I said, surgical procedure that you might give consent to,
Yeah.
and people weigh the risks and benefits of one-time procedures, and then when the benefit exceeds the risk, they'll say, "All right, I'm willing to take this on."
Yeah.
And that's kind of where we are right now. And I think our belief is, over time, over the next few years, we'll generate a body of evidence to hopefully show that the benefit of this one-time procedure exceeds any risks.
Yeah. I am reminded of a book, is it James Forrester's Heart Healers, where he goes into that kind of, the history of, a lot of heart-based surgery and other procedures, and just how much those kind of boundaries were pushed, and then the kind of then the benefits were seen, but just how many, very bold and brave people have basically been, heart surgeons or cardiovascular in nature.
Yeah, that's right. I think there's great examples. I think Andreas Grunzig, who invented the coronary angioplasty,
Mhm.
I think did a lot of pioneering work, even, sometimes self-catheterizing,
Yeah,
to really make discoveries. And so there's lots of stories like this. Yeah.
yeah. Okay. I mean, the other element of ethics is clearly the kind of commercial one, right? Because, one of the challenges for our industry when it looks at the higher priced approaches to medicine is the kind of, the ability to distribute this to everybody. And how do you kind of make this equitable as a therapy?
Yeah. I mean, that's one place we're fundamentally different
Hmm.
from other one and done therapies. I
Yeah,
think when people think about one and done, they think a very high price point, often millions of dollars for that
yeah,
single. And that typically is the case, Mike, because most one and dones to date have focused on rare conditions,
yeah,
addressing rare conditions, so small number of patients. And then second is the manufacturing costs have been quite high, necessitating that combination necessitates a very high price point.
yeah.
We're the exact opposite. We're literally trying to treat the leading cause of death in the world. So it's millions, if not hundreds of millions of patients around the
Mm,
world. So very large indication. Second, our cost of goods ultimately we think is going to be much more manageable. And because our drug product is mRNA packaged in a lipid nanoparticle. So it's RNA packaged in a lipid nanoparticle. This was our concept back in 2016, pre-COVID. Then COVID comes along in 2020, and the COVID mRNA vaccines look just like our drug.
Right.
It's RNA packaged in lipid nanoparticle. Now we know that kind of product can be manufactured at scale very cost effectively.
Hmm.
We think ultimately our drug, maybe 50, 60 milligrams, 70 milligrams that we might give to patients as an intravenous infusion can be manufactured, Mike, for a couple of $1,000s.
Mhm.
So that will give us a lot of flexibility on pricing
Yeah.
and market access.
Yeah.
And so I think we will be in a position ultimately to make this hopefully accessible to the whole world.
Yeah. And you and I have had conversations about the kind of commercial model and, how you might approach this. Does your link to Lilly give you more room for maneuver, or are they going down traditional models, do you think?
Way more.
Mhm.
I'll just point you to Dave Ricks, Lilly's CEO, just was on this like
Yes.
fantastic 2-hour podcast
Yeah.
called Cheek Pint with the Collision Brothers. And, listen to that 30 minutes in.
Mhm.
And Dave starts talking about one and done therapies that are potentially for large indications, and starts talking about, we have a cholesterol lowering one and done in development, and there are issues to solve going forward, safety, but assuming it works, his question is, how would one price that?
Yeah.
You know? And so it's a fascinating answer that he gives. And I think what it said to me, the way he approached it is there's going to have to be innovation here. Just like we've innovated on the technology to date
Yeah.
and gotten it to work in cells, mice, and now patients, I think there's going to be another round of innovation on how to deliver this and how to make it accessible, where you have a product that delivers value with a one-time treatment. And how do that payment, do you have to capture all that value at that initial time point?
Yeah.
Or is there another way to think about distributing that?
Yeah. I think a bit like you guys with this sort of steady pursuit of a goal through different approaches, different technologies. I think that same goal, to make sure people do have access is gonna allow you to innovate some different models, right?
Yeah. And I think that, in some way, I view our approach to cardiovascular disease, specifically atherosclerotic cardiovascular disease, our treatment approach over the last 50 years has been backwards.
Mhm.
This is a disease that is literally the cumulative exposure over decades
Yeah.
of several causal insults. Okay. Cholesterol, blood pressure, and weight/diabetes, probably the three most important in my mind.
Mm.
Okay. And what you wanted, our approach to date has been wait till the patient has very advanced disease,
Yeah.
typically in their 60s or 70s. Typically, they've already had a heart attack,
Yeah.
a consequence of that buildup over time. And then you come in with a sledgehammer to dramatically lower the cholesterol, for example. Okay. And then you'll get some degree of risk reduction.
Yeah.
So, for example, Mike, if you lower cholesterol 40 milligram deciliter for 5 years, starting at a time that somebody's already had a heart attack, let's say average age is like 67 in the US. So you treat them from 67 to 72, 40 milligram deciliter lowering, you get about a 22% risk reduction in the future event.
Right.
Okay. That's good, but not great, right? And so then people talk about residual risk. There's still a lot of risk left. So what's the answer to that residual risk? Well, one answer could be, well, you're missing some other new mechanisms. I need to go find that and give it to the person in addition to the cholesterol lowering.
Yeah.
Right. But to be honest, the answer could equally be just start earlier for the known causal insults. And the human genetics would suggest that actually the answer to the residual risk is just starting earlier.
Yeah.
With cholesterol lowering, for example.
Yeah.
Because that same 40 milligram deciliter lowering, that over 5 years, starting at 67, that gives you the 22% reduction. If you started that much earlier, let's say you're able to maintain that difference for a lifetime,
Mm-hmm.
the risk reduction, Mike, is not 22%, but like 90%.
Yeah.
Modeled by the human genetics. So you could like wipe out this disease.
Yeah.
You're able to get the cholesterol a bit lower and keep it low for a long time.
Yeah.
And so the approach to treatment needs to be inverted,
Yeah.
flipped. You need to have actually more modest effect potentially, but just do it for a much longer time, have it be durable.
Yeah.
And that's really what something like gene editing will allow for.
Yeah.
Right? Have it started earlier, have it durable one time, and have the cholesterol low for a long time. Now, I think the question then is, how is our system set up? What is our system set up
Yeah.
to actually value that? And I think we're gonna have to figure that out, hopefully with,
Yeah.
with
And to see whether, the US versus maybe the UK versus Europe versus, Japan, China, well, how they approach that as well. Because I think some of those countries are going to have a very different, idea of what those models might look like.
that's right.
Whether you're shifting insurers every 3 years or you're sticking with the UK's NHS for a bit longer. I'd be interested. I mean, I hadn't thought about this question, but you look at what's happening with GLP-1s and where they're having their effects, and a lot of it is being discovered now versus pre-hoc. Do you see the same thing happening with the kind of reduction of LDL? Over time, dementia, other kind of ancillary benefits?
Yeah, I mean, the reason that atherosclerotic cardiovascular disease is a leading cause of death is the atherosclerosis that comes from LDL
Mm.
can happen in any artery.
Yeah.
Yeah. So if it happens in the heart arteries, the blockages, that leads to heart attack, leading cause. But if it happens in the arteries to the brain or inside the brain, the major issue is stroke and dementia.
Mm.
If it happens in the leg arteries, amputation and peripheral vascular disease. So there's a range of benefits that can come from LDL lowering. So I think you're going to see that we know that already, to be honest, from the existing cholesterol lowering medicines, but I think we may be able to prove it out even more clearly with a medicine like ours, which is expected to lower and keep it low for a long time.
Yeah. Yeah. Okay. I'm keen to get to the Lilly thing because, this time last year, I had no idea this was going to happen. And then suddenly it's there. To tell me how you approach, conversation like that. You were on your way into, towards approval. And then someone at the scale of Lilly comes along and says that they're interested. How does that conversation go? And, what kind of non-negotiables did you have in the conversation?
Yeah, I think the key thing that I've learned is it starts with the relationships,
Mhm.
you know, and we got introduced a couple of years ago to the leadership team of Lilly.
Yeah.
Ruth Meno, who leads cardiometabolic research, Ken Custer, who at the time had led BD, but now leads the business unit, cardiometabolic, so the commercial side, and ultimately Dan Skravansky and
Yep.
Andrew Adams, who led genetic medicines. That group, I think our technology and the desire to apply this technology to the masses piqued their interest. That gene editing, the application might not be just for rare disease or cancer, but maybe, cardiometabolic disease. So that's kind of where it all started. Then a couple of years, we started working together on a few different projects, one on lipoprotein little a, and another on our lead two programs, PCSK9 and ANGPTL3, where they ended up acquiring rights that Beam Therapeutics had held.
Yep.
So in that context, they kind of dipped their toes in the water and tried to understand what we were doing, having a kind of a front row seat, and got to know the team
Mhm.
and the kind of problem solving that we were doing. And that brought us basically to late last year, around the time of the American Heart Association meeting, when we were chatting about what does 2025 look like, and they were facing a potential decision around opting in to what our lead program at the end of the dose escalation in phase 1.
Yeah.
And they basically proposed, "Hey, what about maybe a larger transaction?" And that kicked off, roughly 6 months of discussions and led ultimately to the acquisition. And I think our board, and including myself, felt this was such the right move because, our ambition is large. We wanna develop this medicine and have it available to a large swath of the population. We have done a lot of the de-risking and getting it to phase 1, early phase 1 data, but we had data in about 14 patients back in
Right.
April. Still a long road to go, you know? Roughly probably another half a dozen years,
Mhm.
probably another 1.5 to 2 billion of capital to get this to approval, and a myriad of risks ahead, different risks,
Yep.
clinical risk, regulatory risk, and commercial risk ultimately,
Yep.
right? Which was a big, still thing hanging out there.
Mm.
And what was really gratifying was Lilly is ready to take on that risk, because they see the potential, right? Because if this whole thing works, it has the potential to change the way we practice medicine.
Yeah. Yep.
And, and they have, I think they're in a privileged position right now. They, and now we, I'm now part of Lilly. We have the privileged position of being able to take the long view.
Yeah.
Right.
Yeah.
Currently, the wonderful innovation, led by Roots Group, in cardiometabolic research in terms of tirzepatide and eloralantide and orforglipron.
Mm.
And so the weight loss medicines, you know, are transforming the way we approach that disease, and they're getting actually rewarded for that.
Yep.
And with that capital, they're looking for, okay, what are the next transformative advances? And can we make some bets on those? And so we at Verve are incredibly grateful that they chose to make that bet on us.
Yeah, and you've said, in the past, Verve was built to disrupt chronic care models. So, is he sharing that vision across Lilly now?
Yeah, I think our vision has always been that for targets of high conviction that are proven mechanistically to cause disease,
Hmm.
PCSK9 being one of them, and ultimately, hopefully, LPA being another, that there will be a range of modalities, like stacking of modalities against a given target.
Yeah.
So, for example, for LPA, Lilly has a pill. Lilly will have has an SI in phase 3. So, a daily pill, once a year or twice a year injection, and then working with us for gene editing.
Mhm.
Imagine a similar setup for PCSK9 as well, and maybe some of the other targets, so that these options will be there for providers, payers, and patients.
Yeah.
And that's, I think, so the one and done will sit beside, I should say, the chronic care model. And, we've talked about this. We've done some surveys we and others have done surveys of patients who require lifelong cholesterol lowering and have put forward a future treatment landscape of a daily pill, twice a year injection, twice a month injection, and one and done.
Hmm.
And the results are very interesting. And they speak to, I think, what would be the future. About 40% prefer a daily pill.
Yep.
About 14% prefer a twice a year injection. About 4% prefer twice a month injection. And a full third, already at this stage, prefer a one and done,
Nice.
a
Yeah.
one and done gene editing option. And so let's offer that full technology stack to
Yeah.
each patient.
Yeah. And that faith, I guess, will only increase as you go. Absolutely. Yeah. I'm guessing, your rise to where you've got to has been pretty rapid. When the broader ecosystem hasn't, done so well in the last 2 or 3 years. It seems to be getting better, but if you could edit one of the genes in the ecosystem, where would you be putting your attention at the moment? Where do things need to improve?
Well, I would take a step back and I kind of think about what has enabled the Verve story,
Hmm.
you know? And how is it that somebody can come in with a new idea that, is a little bit, again, viewed as a little bit out there not that long ago, and then raise the sufficient necessary capital, work with our ecosystem to get to where we are now.
Yeah,
There's a series of handoffs that have happened, right?
yeah,
And when I look back, I think, again, not to be too jingoistic, but I think this can really only happen in the United States
hmm,
and right now. And I'd like for us to think about what is it about our system that has allowed this to happen and figure out how to preserve it,
yeah,
you know? And at a simplest level, it is really incentives driving innovation, you know? And there have been to date ample incentives at each step for capital to come in and allow for that innovation to happen. And there's a series of handoffs
yeah,
from VCs to the next round of investors, now ultimately to large pharma, right? And, but all of that, what's been allowed to happen over the last decade, a lot of that is at risk, I think, you
yeah,
know? Even going back to the earliest part of this handoff, which is from academia to
yeah,
the company, in terms of research funding that enable the discovery and perfection of CRISPR technology.
mm,
That's at risk. The next phase in terms of capital flow in the US into biotech is at risk,
mm,
largely from competition from China,
yeah,
you know. The regulatory apparatus, the regulatory certainty that is necessary for this flow to happen is at risk because there's a lot of capriciousness right now, arbitrary nature to various, to the regulatory apparatus, particularly in the United States,
yeah,
right? And then, of course, the last piece of the puzzle, pharma and commercial and being able to be incentivized to develop new medicines and ultimately get paid for that. That's also in a bit of a shifting landscape.
yeah. Do you feel
So
optimistic, or
sorry?
do you feel optimistic about where it's going, or
I'm very optimistic,
yeah,
but I think we need to take a hard look. I'm optimistic because of my journey. I don't think I could have done this anywhere else in the world, particularly as an immigrant
yeah,
coming to the United States at the age of 9, and really just, making all this happen.
yeah,
But, is an immigrant from India coming today at the age of 9, in, you know, 40 years gonna have that same opportunity in the United States?
yeah,
I don't know. I don't know.
okay,
But I think we need to take a look at what's made it all work so far and try to preserve those key elements.
yeah. And it's going to be easy to unravel some of those ecosystem things, right? And you say, I think the presence of China, I think, is still taking people by surprise. Quite what threat we're facing from that desire they have to commercialize globally now. I think, which is the main thing that's different.
Absolutely. And their innovation, they have stepped up their innovation, right?
Mm,
And now kind of the go-to, as about roughly a third of all capital for
mm.
early stage deals went to China in the last 18 months.
Right. Yeah. Okay. Sec, I know we need to close. So thank you for everything we've discussed so far. But maybe a kind of a big question at the end. What's one question about your work or about Verve's work that you wish more people had asked you either, this year or before now?
Well, I think it is this item that I mentioned. Is it like what is it that enabled this to happen?
Yeah.
Particularly for essentially what was a risky bet, and then having some success,
Mm,
early success, and I think that we really need to take stock of this U.S. system and try to preserve all those good things that allowed something like Verve to flourish so far. And I keep coming back to incentives. It's really incredible to think about. This is what makes the world go. Incentives.
mm,
he cannot forget that. And any time that you try to do it in a slightly different way, you don't get as innovative results.
Yeah. Yeah. Now, I've written a lot about planned serendipity. You want good things to come out at the end. You can't just put it all out there as hope or luck. There needs to be a bit more planning in the serendipity. So it's interesting that you say that. But Seth, I know I always enjoy our conversations, and this has been eye-opening. Congratulations on what you've done, and I'm looking forward to the kind of next milestones that are out there. So thank you, Sek.
Thank you so much. I really appreciate you having me on the program.
Really enjoyed it.
That's it for this week's episode of Idea Collider. To continue the conversation, visit our website at ideapharma.com. Follow us in your favorite podcast app so you never miss an episode. Don't forget to rate and review us on Apple Podcast. Until next time, I'm Mike Rea, wishing you great success.